Oxygen transport. Is our focus for this morning's lesson. And what we're looking at here is how hemoglobin is so very, very, very perfect for transporting oxygen. There's a few features that just make it the best ever and really efficient, which is something that we need when we're transporting oxygen throughout the body. That oxygen's got to get from your lungs to every single cell, even the ones at the end of your fingers and your toes. So we need to be able to carry that oxygen there, and we need to be able to release it as soon as we need to use it in the cells. So, transport of oxygen. We need to start out with that all-important reaction of Hb plus 4O2, reversible reaction to HbO8. So we're getting a little bit more into the chemistry in this side of things, and it's important that you are happy with that chemistry. So what is each of these things? Well, this is your hemoglobin. Here is your 4 oxygen molecules. And then what we get on this side is our oxyhemoglobin. So it's that oxyhemoglobin where we're now carrying our oxygen. And it's reversible, which means we can go hemoglobin plus oxygen goes to oxyhemoglobin, or we can go in the other direction. Oxyhemoglobin can dissociate to form the oxygen molecules and the hemoglobin. So in the lungs, we have the combination, the pickup. In the tissues, we have the breakdown and the loss. So we're going different directions in different places depending on what's needed and what's happening. This is really important, okay, that we can go in both directions. And the other aspect of this is something that we call affinity. So if you have an affinity for something, then you like it and you're attracted to it. So I have an affinity for hot chocolate, most definitely, not under debate. Okay, hemoglobin has an affinity for oxygen. If we now look at how we know this, okay, and how this works into what we need to know, when we think about it, we've got 4 oxygen molecules, right? So what happens is that those oxygen molecules, as they bind, actually affect the hemoglobin molecule, increasing the affinity, so making it easier for the next one to bind. So we have our hemoglobin molecule. Remember, hemoglobin is your 4 polypeptides, 2 alpha globin, 2 beta globin. Each one of these has a heme group. Remember that protein structure is really specific, and as soon as you change something, then it can have a knock-on effect on the rest of the structure. So as soon as the first oxygen comes in and binds, this causes a slight distortion in the rest of the molecule because these 4 polypeptides are bonded together. We've got quaternary structure going on here. They're not just sort of loosely associated, they're actually bonded together. So the first one is going to distort the molecule slightly. That then makes it easier for the next one to bind. This causes a further distortion because we're changing the shape. When we bond, we change shape, we change interactions, we change molecule, we distort. Okay, so that makes it easier for the next one to bind. Yet again, you have that same process. The third one bound then creates another distortion, which makes it even easier to bind the 4th one. So the beginning of oxygen, it's easier to get more and more oxygen. So the more oxygen you pick up, the easier it is to pick up all your oxygen. So how does this actually relate to some numbers? We said we're going to get more technical with this. Up to about 2 kilopascals of oxygen partial pressure. Then you only have 1 oxygen molecule per hemoglobin. Let's make that a more obvious one. At higher partial pressures, you get a steeper increase because we've got this distortion going on. This is what leads to our S shape for our saturation curve for hemoglobin, which we're going to look at just now. We're going to draw the curve and actually look at it in a bit more details. But it's quite important also to think of this in context. We talk about 1 hemoglobin molecule, right? 1 hemoglobin molecule getting 1, then 2, then 3, then 4. But remember that you've got lots more than 1 hemoglobin molecules in each red blood cell. And then you've got a lot of red blood cells going past each alveolus. So we're talking here on sort of cross scales as well. So it's important to remember that when we're talking about up to 2 kilopascals of oxygen partial pressure, we have 1 oxygen per hemoglobin. That is per hemoglobin, but we've got lots of hemoglobin. So they're all carrying 1. Then they all get more. So what does this look like, this saturation of hemoglobin graph? Okay. This is not going to be a very precise graph, but it'll give a good idea. So we go from, well, up to 100 from 0, and then we usually go up to about 14 on this side. And here on a vertical, we have percentage saturation of hemoglobin by with, you're saturated with something, oxygen. Okay, and at the bottom here, we have partial pressure of oxygen. That's just a fancy way of saying the concentration of oxygen. Okay, this is measured in kilopascals because there's a pressure. Okay, so when we have no oxygen, we have no saturation of hemoglobin. We said then to about 2, we get about 2 goes up to about 1 per hemoglobin. So 25% saturation. Then we go steeper, and then we level off now because we fully saturated. So when we have 100% percentage saturation, this is fully saturated. Every hemoglobin has got 4 oxygen molecules. Here we have no oxygen molecules. Halfway, we would have 2 oxygen molecules per hemoglobin molecule. Here we would have 1 O2, and here we would have 3 O2s. So we can see we get the first one and we're going up really steeply. So think about now, where have we got this? What are we thinking about? Okay, we can understand it in terms of saturation and number of oxygen molecules bonded, but we can also understand this about where we are in the body. So if we look on this side, partial pressure of oxygen, thinking about that as concentration of oxygen. Where are we going to get low and where are we going to get high? Down here in the low is going to be our respiring tissues, our muscles, because we're using that oxygen there. Up here on this side, high concentration, high partial pressure of oxygen is going to be your lungs. Because there you've got the fresh air that's being breathed in, so it's still got a high level of oxygen. What this means is it links back then to how saturated we are. What we're saying is that in the respiring tissues, we're going to have no saturation. Low partial pressures of oxygen, we're going to get rid of that oxygen. Whereas high partial pressures of oxygen in the lungs, we're going to be saturated. And unsaturated. So we're looking here at sort of different places, different regions in the body, but we're doing it by looking at, in that area, what is the partial pressure of oxygen. So if we find a place in the body which has a partial pressure of whatever this partial pressure is, and we go up and we would look then, oh, okay, well, this is going to be 75% saturated. So a little bit of oxygen lost, okay, donated to the tissues, but the bulk of the oxygen is still with our hemoglobin, ready to transport somewhere else where it's needed. Are we happy? Cool. So we understand now that hemoglobin can pick up and hemoglobin can drop off, and we have this understanding of where there is a high oxygen concentration, then hemoglobin is going to pick up the oxygen. Where there's a low partial pressure of oxygen, then the hemoglobin is going to lose its oxygen, which is great. But there is another element to this story. Okay? There is something that makes this even better, even more efficient. And remember, efficiency is always a good thing when we're talking about processes. So let us have a chat about the Bohr shift. The Bohr shift is where the questions come in. It's this understanding of how things can change. And the brilliant thing about this is that we've considered only partial pressure of oxygen. If we now consider partial pressure of carbon dioxide, we can make this system even better. Think about it. High levels of respiration. These are going to produce lots of carbon dioxide, right? Requiring more oxygen. So if we can get some sort of interaction between carbon dioxide and this letting go of the oxygen, then we can make this even more efficient. And that's exactly what happens. So we have carbon dioxide and it comes out of the respiring cells. It diffuses into the blood plasma. It diffuses into the red blood cell cytoplasm. That's a whole lot of cell membranes to cross, okay? But it can do so. So think about this in terms of transport as well. Make links back to transport across cell membranes and those sorts of fun things. Right, we get into the red blood cell cytoplasm. Then we have carbon dioxide. We add to that some water. We add to that a reversible reaction and we get H2CO3. So for those of us who have not got so much of a chemistry bent, CO2 we're happy with, H2O we're happy with. There's an enzyme involved here because we are biologists. Carbonic anhydrase. Everything in biology is enzyme controlled. Carbonic anhydrase makes carbonic acid. So you can use the names of the different things to help you remember. Okay, now acids we say, but acids have like hydrogen ions in them. Well, let's get there. The H2CO3 can dissociate. Okay, that means it doesn't have to stay as one molecule. It can sort of break up into two ions. Our hydrogen ion that we want for our acid, and what's left? HCO3 minus. Because if that's a plus, that must be a minus. This is your hydrogen carbonate ion. Remember that 85% of your carbon dioxide is carried in your blood plasma as hydrogen carbonate ions. So these guys are really important. Okay, the other 5% of your carbon dioxide just stays in your blood plasma, is diffused, and then you've got 10% that forms carbamine or hemoglobin. A lovely long word as well. But for the moment, we're sticking with what's going on in the Bohr shift. So this guy, this hydrogen carbonate ion, he can diffuse into plasma and carried. So in the lungs, see how all of this is reversible, okay? In the lungs, he can combine with a hydrogen ion, he can make combine with a hydrogen ion, make carbonic acid, which can go back to carbon dioxide and water for exhalation. But for the moment, he's diffusing in the plasma because we're talking about our high carbon dioxide. So we've created these hydrogen ions. And this is where the story gets interesting. Now, we have said that hemoglobin has a high affinity for oxygen, but hydrogen is a little bit interesting as well. Hydrogen and hemoglobin have a relationship going on. Okay, this hydrogen ion combines with the hemoglobin. To form HB, which is hemoglobinic acid. So the hemoglobin actually sort of says, woohoo, I don't want this oxygen anymore. I want that hydrogen ion. Remember, there's all sorts of charge issues going on with proteins. So this nice and stable oxygen molecule is not as attractive as this hydrogen ion. So in order to bind with the hydrogen ion, the hemoglobin has got to give up its oxygen. Think about it. Carbon dioxide has led to the formation of the hydrogen ions in the tissue. Okay? The hemoglobin wants to bind with the hydrogen ion, so it releases its oxygen in the tissue. So we have high respiration, producing carbon dioxide, which produces hydrogen ions, which then releases oxygen in that self-same tissue, which means that we can then continue that high respiration. Aerobic respiration is the best, requiring oxygen. So, in the tissues, we have an increase in our oxygen release. So why is this called a Bohr shift? And it's because it shifts that whole curve that we drew previously to the right. It changes that level of affinity and the saturation that we have at different oxygen partial pressures. So let's just make a note here. So we have respiring tissues. So then we have carbon dioxide, we have carbonic acid, we have hydrogen ions, we have hemoglobinic acid, and therefore we have increased oxygen released. So we're increasing the efficiency of this process, this release of the oxygen in the tissues where it is needed. So let us now have a look at actually what that Bohr shift looks like on a graph and how that can then relate into questions. So we shall start again with our curve like we had last time. From the bottom, slowly, fast, and then we're up at high partial pressures. So this is again percentage saturation of hemoglobin with oxygen. all the way up to 100% saturation from 0 known to all 4 present. Then let's take it over a bit to 14, and here we have partial pressure of oxygen. So we're looking at the exact same saturation curve. Now, what we've said is that we shift to the right. So we're going to draw another curve that is just shifted to the right. We don't have to get involved in the mathematics of it, just the appearance. And now we need to think. This curve is what happens in high carbon dioxide. So this one here is what happens in low carbon dioxide. What does this mean? If we have high carbon dioxide, when we think about our respiring tissues, our tissues are going to be on this pink line. Okay, so our respiring tissues, low partial pressure of oxygen, high partial pressure CO2. Okay, so low oxygen, high CO2. Our lungs are the opposite. Lungs have high oxygen, low CO2. Oopsie, let's be consistent and make it fit. Low CO2. So when we're in the lungs, we'll be on the black curve at the top. When we're in the tissues, we'll actually fall on the pink curve at the bottom. Okay, great. But now what if we're somewhere else? Or what if we're comparing different situations? How does this actually make sense? So the way I like to do it is to say, okay, so at a particular partial pressure of oxygen, what is the difference between these two curves? So at our particular pressure of oxygen, when we have high carbon dioxide, we have much fewer oxygens. Than if we have low carbon dioxide. So here we have a higher saturation, and down here we have a lower saturation. So if we are comparing saturation in different areas of the body or in different organisms, we might need to take it a little step further, because sometimes we relate this to affinity. If you have higher or lower affinity for oxygen, okay, then it means that your hemoglobin is more attracted to oxygen or less attracted to oxygen. So you can see how this now starts to relate to the same thing, because when we have this high carbon dioxide, we're effectively decreasing our affinity for oxygen because of the presence of those hydrogen ions. The hemoglobin wants to bond to the hydrogen ions instead of the oxygen. So lower saturation, we can interpret this also as lower affinity. And therefore higher saturation also converts to higher affinity. Understanding it like this can really help you when you get those multiple choice questions with those shifted graphs and you've got 4 or 5 lines and now you've got to figure out who's who in the zoo or what they're representing. So always remember that your high carbon dioxide is shifting you to the right. Higher carbon dioxide is going to be lower saturation in oxygen, okay? And that can also be linked to lower affinity for oxygen. And it all makes sense if we pull it all together and we get even more colorful, because this is just asking for colors. These guys are what you're going to want if you have high carbon dioxide, because when you have a higher carbon dioxide level, you are indicating a higher oxygen demand. So we have a higher CO2 level, we have a higher oxygen demand. Therefore, we want to be able to have a lower affinity, a lower saturation, because that means we are releasing that oxygen. So those oxygen molecules are no longer bonded to the hemoglobin, and they are free to be used then by the cells in respiration. So it's very important that you've got an idea. This shift to the right is the Bohr shift. Okay, and there you go. You underline him with an arrow, and that helps you to remember that your Bohr shift always goes to the right. Okay, it's got two important things to remember also. One is that you're going to release oxygen, and the other one is that you're not going to have all these hydrogen ions lurking around, because we don't want our red blood cells to be getting all acidic, because now they've got these hydrogen ions from that carbonic acid. The hemoglobin is binding with those hydrogen ions and keeping our pH where it should be. So there's two knock-on effects there, but the big one to really get a handle on when it comes to understanding these graphs is that difference between the saturation and how that links to lower and higher affinity, and how that can help you decode and understand those multiple choice questions that love to crop up.